Hello and welcome to EaseGenetics. I'm Dr. Kaveh Zargari, and with 28 years of experience teaching genetics to students at every level, from bachelor's to PhD, I'm here to help simplify the fascinating world of genetics for you. Whether you're a student preparing for exams, a researcher brushing up on key concepts, or just someone curious about how life works at the molecular level, you're in the right place. With Linkage 101, today's topic is two-point linkage. Together, we'll explore how genes on the same chromosome interact, how to calculate recombination frequencies, and what this tells us about genetic distance. So grab a notebook and let's make genetics a little easier, one concept at a time. Thomas Hunt Morgan was the first scientist to discover genetic linkage, showing that genes on the same chromosome are inherited together. His experiments with the fruit flies revolutionized genetics and earned him the 1933 Nobel Prize. What is linkage in genetics? In genetics, linkage refers to the tendency of genes located close to each other on the same chromosome to be inherited together during meiosis, rather than assorting independently. This is because genes physically near one another are less likely to be separated by recombination, which occurs during the formation of gametes. Let's see how it looks. Take a look at this visual. Here we have three schematic chromosomes, each with gene markers labeled as A, B, D, E, and F. First, focus on the first chromosomes labeled 1 and 2. Notice that chromosome 1 carries the gene A, while chromosome 2 carries the gene B. These two genes are on separate chromosomes, meaning they are inherited independently. This is an example of independent assortment, one of Mendel's key principles. Because they're on different chromosomes, the inheritance of one doesn't affect the inheritance of the other. Now let's look at the third chromosome, labeled 3. Here we see three genes, D, E, and F. Unlike the previous examples, all three are located on the same chromosome. Because they share the same physical space, these genes are considered linked. What does this mean? It means they tend to be inherited together, as the chromosome they are on is passed down as a single unit during meiosis. In conclusion, all genes on a single chromosome form a linkage group. This simple yet powerful concept of independent versus linked genes is fundamental to understanding how traits are passed down through generations. First, let's see what a crossing over is and what the result of a crossing over will be. At the left, we start with two homologous chromosomes, one set in green and one in purple. They're a matching pair, but with different genetic material, carrying a combination of dominant A and B genes on one chromosome and recessive A and B genes on the other. Now, in the middle of this diagram, things get interesting. We see that crossing over occurs. This is the chromosome where sections of genetic material swap places between the two homologous chromosomes. Those segments cross, twist, and then break and reattach to the opposite chromosome like a perfectly synchronized routine. After crossing over, we're left with our recombinant chromatids, shown at the right. Here's the twist clearly. Now we have new combinations, one chromatid with a dominant A and recessive B, and another with a recessive A and a dominant B genes. Here is an example of how recombination works. In humans, hair and eye color traits are determined by the combination of genes inherited from the parents. Hair and eye colors are influenced by multiple genes, some of which are linked, meaning they are located close together on the same chromosome and often inherited as a set. So most individuals have dark hair and dark eyes or blonde hair and blue eyes. Dark hair and dark eyes are usually dominant in genetic inheritance, meaning they are more likely to appear in the next generation, while blonde hair and blue eyes are recessive and often only appear when two copies of the recessive gene are inherited, one from each parent. Now, let's take a look at this family of four. On the top left is the father, who has dark hair and dark eyes. Beside him is the mother, with blonde hair and blue eyes. Below them are their two children, the son, who has dark hair and blue eyes, and the daughter, who has blonde hair and dark eyes. The father's dark hair and dark eyes suggest he carries dominant genes for these traits. The mother's blonde hair and blue eyes indicate she carries recessive genes for both traits. The son has dark hair and blue eyes. This shows he inherited the dominant dark hair gene from his father and the recessive blue eye gene from his mother. The daughter, on the other hand, has blonde hair and dark eyes. She inherited the recessive blonde hair gene from her mother and the dominant dark eye gene from her father. In this family, the children's traits suggest some recombination occurred, where linked genes were shuffled during meiosis. According to gene linkage, there are two types of gene arrangements: cis configuration and trans configuration, along with their implications for gamete production. On the left, we have the cis configuration. In this setup, the two dominant alleles A and B are located on the same chromosome, while the recessive alleles A and B are on the homologous chromosome. This arrangement leads to parental gametes being the same as the original chromosomes, which are both dominant AB and recessive AB. However, if recombination occurs, recombinant gametes are produced, resulting in new allele combinations, one with dominant A and recessive B, and the other with recessive A and dominant B alleles. On the right, the transconfiguration is shown. Here, the dominant alleles A and B are on opposite chromosomes, paired with the recessive alleles B and A, respectively. In this case, the parental gametes reflect the trans arrangement. Recombinant gametes following recombination will produce dominant A and B and recessive A and B, effectively flipping the pattern seen in the cis configuration. The fundamental difference between linked genes and independent genes is specifically focusing on the ratio of gametes produced in a dihybrid cross. What makes linked genes differ from independent genes? The answer is it's all about the ratios of the gametes. When genes assort independently, as per Mendel's law of independent assortment, the dihybrid produces gametes in a 1 to 1 to 1 to 1 ratio. When genes are linked, their gametes deviate from this 1 to 1 to 1 to 1 ratio. The ratios shift because linked genes tend to stay together during meiosis, reducing the frequency of recombination. Here's the new pattern for linked genes. Parental gametes occur more frequently while recombinant gametes occur less frequently. This unequal ratio is a hallmark of linkage, where recombination events are rare due to the proximity of the genes on the chromosome. Let's compare the gametes produced when genes are independent or linked in details. First, we focus on independent genes and how they produce gametes with equal probabilities. Here we have a dihybrid individual. The slash indicates that gene A is on one chromosome and gene B is on another. These two cases illustrate how independent assortment ensures that all possible gamete combinations are produced in equal proportions. In case 1, during meiosis, the chromosome assort independently. One gamete may inherit the dominant allele A along with the dominant B, so the other gamete will inherit the recessive alleles A and B. In case 2, alternatively, one gamete may inherit the dominant allele A along with the recessive allele B, so the other gamete will inherit the recessive alleles A and dominant allele B. The result is 4 gametes produced in a 1 to 1 to 1 to 1 ratio. Now we focus on linked genes and how they produce gametes. Let's look at cell 1 on the left. Here we see a pair of homologous chromosomes with genes A and B in a cis configuration. Notice how the alleles for A and B are in the same order on each chromosome. This is because there's no crossing over happening here. Since the genes are physically close and not separated by crossing over, they're inherited as a unit. Now let's look at cell 2 on the right. This time, crossing over has occurred. You can see that the alleles for A and B have been swapped between the chromosomes. To understand the difference in gamete ratio production, consider 2 scenarios, each with 2 cells, one with independent genes and the other with 2 linked genes. On the left, we see 2 cells, each producing 8 gametes in total. All 4 types of genotypes are made, with 2 of each type. As a result, the final ratio of gametes is 1 to 1 to 1 to 1. On the right, assuming a cis configuration for the parent cell, we observe 2 cells with linked genes. As mentioned earlier, crossing over between the 2 genes may or may not occur, so we must consider both possibilities. In the left cell, crossing over does not happen, resulting in parental gametes. Crossing over occurs in the right cell, producing all 4 genotypes. In total, these 2 cells also produce 8 gametes. However, unlike the first scenario, we see 6 gametes each with parental type genotypes and 2 gametes with recombinant type genotypes. As a result, the gamete genotypes are no longer in a 1 to 1 to 1 to 1 ratio. Instead, the parental types are more frequent than the recombinant types. Now, imagine hundreds of thousands of cells undergoing meiosis. Depending on the distance between the 2 genes, the proportion of recombinant genotypes can range from 0% to less than 50%. If the recombinant gametes make up exactly 50%, it indicates that the 2 genes are either independent or far enough apart on the chromosome to behave as if they are independent. To solve this, we can introduce a 3rd gene between them to reduce the genetic distance to less than 50 map units. This concept will be explored in detail in my next video on 3-point genetics mapping. For example, what if we have 50 cells? Each cell represents a meiotic event. Assume in 49 of the cells there is no crossing over. This means that genes A and B are inherited together as a unit. However, in one of the cells, crossing over occurs between the genes A and B. This breaks the linkage and creates new combinations of alleles. By measuring recombination frequency, we can estimate the distance between genes on a chromosome. This information is crucial for creating genetic maps. Now let's calculate the frequency of recombinant gametes. We have 2 recombinant gametes out of a total of 200 gametes. So the frequency of recombinant gametes is 2/200, which simplifies to 1/100 or 1%. This means that 1% of the gametes produced in this scenario are recombinant. Remember that the frequency of recombinant gametes is proportional to the distance between the genes. Considering that the likelihood of recombination occurring between genes during meiosis determines the distance between two genes, the number of offsprings produced is highly important. The more offspring, the more accurately we determine the distance between the genes. To illustrate the distance between these two genes, we can draw an imaginary section of the chromosome. By selecting 2 points, we represent the two genes on the chromosome and indicate the distance between them. Note that a map unit, also known as centimorgan, is a unit of measure for genetic distance. One map unit corresponds to a 1% recombination frequency between two genes. This means that on an average, genes that are one map unit apart will be separated by crossing over 1% of the time during meiosis. There are also other scenarios regarding two linked genes. Let's take a look. When crossover occurs between sister chromatids, the outcome is typically genetically inconsequential. Here's why. Sister chromatids are identical. They carry the same genetic information because they are the result of DNA replication during the S phase of the cell cycle. Therefore, exchange between sister chromatids does not alter genetic composition. Even if crossing over happens between them, the alleles being exchanged are the same. Therefore, no new combinations of alleles are produced, and the overall genetic content of the chromatids remains unchanged. Now here, two crossover events occur between the two genes A and B. Despite the crossing over, all four resulting gametes on the right are parental types. Two with the green chromosome, one completely green, one partially purple, carrying dominant alleles A and B, and two with the purple chromosome, one completely purple, one partially green, carrying recessive alleles A and B. No new allele combinations form due to the even number of crossovers. So to see recombinant types, the number of crossovers between two genes must be odd. As you can see here, when crossing over occurs outside the range of two genes on the same chromosome, it also does not affect the linkage or inheritance of those two genes. The two genes remain in their original positions relative to each other. The gametes will retain the parental combinations of the alleles for these two genes, and there is no recombination between the two genes. These events produce parental type gametes, which accumulate with those from crossing over, increasing their overall number. Let's get practical with an imaginary example. We have a plant with red flowers and tall stems. Red flowers are dominant over white, and tall stems are dominant over short ones. To analyze these traits, we'll use uppercase A and lowercase a for flower color and uppercase B and lowercase b for plant height, dominant and recessive respectively. We'll test cross our red tall plant with a white short plant of known genotype, pure recessive, which can only produce one type of gamete AB. What would the results be? If these are the results, 25 red tall offspring, 25 red short offspring, 25 white tall offspring, and 25 white short offspring, we see a 1 to 1 to 1 to 1 ratio of phenotypes. This tells us that the two traits, flower color and plant height, are assorting independently as each is 25%. Also, as the white short parent can only contribute recessive alleles A and B to the offspring, we see white short offspring, and we understand that the red tall parent must also have contributed recessive alleles for both genes. Therefore, the genotype of the red tall parent must be heterozygote. But what if these were the results? In this case, the results will lead us to a different conclusion about the genes involved. As you see, the offspring includes 40 red tall, 10 red short, 10 white tall, and 40 white short plants. Unlike the previous example, we don't see a 1 to 1 to 1 to 1 ratio. Instead, we have two phenotypes, red tall and white short, with higher numbers and the other two with lower numbers. This deviation from the expected ratio suggests that the two genes for flower color and plant height are linked. The two phenotypes with higher numbers, red tall and white short, are, as mentioned before, parental types. They represent the original combinations of alleles present in the parent plants. And the two phenotypes with lower numbers, red short and white tall, are recombinant types. They are the result of crossing over during meiosis. Since the white short parent can only produce recessive AB gametes, the red tall parent must have contributed either dominant AB or recessive AB gametes to produce the parental types. Therefore, the genotype of the red tall parent is heterozygous. As you can see, we can conclude that in this case, the linked genes are in the cis configuration, meaning that due to the parental types of the offspring, the dominant alleles A and B are on the same chromosome, and the recessive alleles A and B are on the other homologous chromosome. The results may also be like this. Here, as mentioned in the previous case, the two genes are linked, but their configuration is different due to the offspring's parental types. We can conclude that based on the parental type offspring, red short and white tall, the red tall parent genes are in the trans configuration. Now the percentage of recombinant offspring is calculated as you see. This 20% recombination frequency corresponds to the genetic distance between genes A and B, which is 20 map units or centimorgans. We can also represent the genetic distance visually by drawing an imaginary chromosome segment. Here, the two genes are marked on the chromosome with the distance between them labeled as 20 map units or centimorgan. Thanks for watching. I hope you found this video helpful and that it made things a little clearer for you. If you're preparing for an exam, I wish you the best of luck. You've got this. Don't forget to like, comment, and subscribe for more videos like this, and feel free to ask any question in the comments. See you in the next one.